Fantastic. Good to
Yeah.
see you, Julie.
Nice to meet you or see you again. Sorry. It's Friday
Good.
afternoon. My brain's already starting to unwind. But my colleague, Reenal Gandhi, is online as well. And
Hello,
hopefully
everyone.
we're going to, so I'm going to build a little bit on what Maria said, just presented in terms of what we think about at Aptar Pharma about devices. And, for those of you that don't know Aptar Pharma, we are a company that works to develop and design and provide delivery systems across many different indications. And you can see here in the slide that we work, across for inhalation, nasal sprays. We have over-the-counter property drug delivery systems, ophthalmic, and injectable. But today we're going to, obviously we're going to focus on our thoughts regarding nasal drug delivery. And, one of the things that I think that's been, sort of fascinating is that, I talk to some KOLs and clinicians, and they're like, why nasal? Why would you even think about a nasal spray? And so sometimes I have to educate them that, systemic delivery of nasal sprays go all the way back until the 90s. And maybe we have seen the nasal route used for treatment for things like migraine pain with Imigram or sumatriptan, going all the way back to the 90s. We saw some slides earlier today, that talked about the importance of Narcan and Neffy. And so with Neffy being the most current systemically administered nasal spray. And actually, I have, maybe a question to the folks on the team. I saw that on one of the slides, I don't remember who presented it, but you had, Narcan and Neffy as a nose to brain delivery system, and which is actually something that I don't actually consider. I think that Narcan, naloxone, and epinephrine are being absorbed from the respiratory region in the nasal cavity into the systemic circulation, and then crossing the blood-brain barrier through the more traditional route. But I'd love to know otherwise, if you think that there actually is a nose-to-brain, route for those products. Maybe you can put that in the chat, or we can talk about it when there's questions. And so, sometimes I also need to, convince people that nasal sprays actually are a viable dosage form. And this just shows the market growth of nasal sprays compared to other dosage forms, and that they're actually growing faster, even though this is a niche within our drug delivery systems. And what I think makes nasal sprays so exciting is that, for all the very same reasons why some of us are on this call today, is that the nasal route can be used potentially to deliver drugs for unmet needs in Parkinson's, Alzheimer's, and more, as you can see here at the bottom of the slide. And so wanted to share some of the work that Aptar has been doing as well, to try and understand how drugs are being absorbed via the nose to brain route, or trying to provide more body of evidence that nose to brain is happening. And this is a study that we did with Lovelace Biomedical in New Mexico, where we used their colony of macaques to deliver sumatriptan in different modalities. So we had 4 arms in the study. We administered a nasal powder of sumatriptan, a nasal spray, an aqueous nasal spray form of it, and a nasal spray using an enhancer, using a permeation enhancer, as well as sub-Q. And so we quantified the drug in plasma and the drug in CSF. And I'm not going to show you those slides for the sake of time today, but, using, the assumption that the subcutaneous route only entered the brain using via the blood-brain barrier and not nose to brain, we can calculate a ratio of the area under the curve of the CSF to plasma. And what you can see here on the right-hand side is that, for the nasal powder and the 2 nasal sprays, we have a higher CSF to plasma ratio, which, again, it suggests that there is a pathway, a nose to brain pathway for sumatriptan into at least into the CSF. Whether it goes deeper into the brain or not, we can't say, but we know at least that seems to indicate that there is that pathway there. And interestingly, the powder performed fairly well compared to the nasal spray with the enhancer. And so, we've taken some of this work further. Is there a question?
No, just someone joined.
Okay. So we've taken this work further through supporting Dr. Suzanne Kraft at Wake Forest University. Dr. Kraft is currently conducting clinical studies on intranasal insulin for improving cognition in patients with MCI and Alzheimer's. And she's been committed to trying to understand, the mechanism of transport of insulin into the brain. So her first step in doing that was to conduct a dosimetry study in adult vervet monkeys using a radio-labeled insulin molecule. So you can see that the tag was actually attached to insulin. So we know that the movement of insulin with the radio label is the actual transport of insulin. So in the study, she did a series of PET CT scans with the animals and administered the radio labeled insulin using our CPS pump, which is one of our multi-dose nasal sprays. And so you can see here in the graph when we look at the uptake of the radio label versus different regions in the brain, we know, or we can see, that insulin is showing up in different regions. The amygdala, the cortex, the cerebellum, the hippocampus. And as we've talked about, earlier in the day, that sort of indicates that we have transport potentially along the trigeminal nerve as well as the olfactory nerve. So, I think this is an exciting study that just further adds to the body of evidence, at least in animals, that we have the nose to brain transport. And so Dr. Kraft is continuing this study and doing a very similar study design in humans. And so we're looking forward to seeing those results in Q1 or Q2 of next year. I think it'll be a very important study to actually look at the transport of the molecule into the brain rather than looking at a pharmacodynamic parameter. And we're happy to be, supporting Dr. Kraft. And so, transitioning a little bit more now into some of the nasal spray considerations. So in addition to some of the parameters that, Maria talked about in terms how the patient actuates the spray, how they insert it into their nasal cavity, what angle they make, we also think a lot about the formulation, whether it's a liquid or a nasal powder. I'm not going to go through all the points here on this slide, but we know that by changing the viscosity, we can change the spray performance. And we saw how, changing viscosity in Maria's slide changes the shape of the spray. And so knowing the interaction of the formulation with the device is important for helping us understand where the droplets or the powder particles may deposit in the nose. So we look very closely at spray pattern, which is the shape, and the plume geometry, which is the angle of the spray, and droplet size in our device development, as well as when our customers are working with the formulation and to help them pick the right device for their clinical trials. And so, some of the things that we have, learned along the way is that, I think it's been stated here today that in the humans, the area within the nasal cavity that represents the olfactory region is quite small. It's only about 5% to 10% of the total volume of the nasal cavity. So we need to think about targeted drug delivery. And so we have done studies with in vitro nasal casts, which are validated nasal casts to look at deposition in the olfactory region. And, our studies show, at least from an in vitro standpoint, that we need a more narrow plume angle, maybe around 20, 30 degrees to target that olfactory region. When we think about the size of the droplets, we want them to be larger than 10 microns because we don't want them to bypass the nasal cavity and deposit in the lung, because that sends a safety signal to regulators when you have lung deposition through the nose. And so, we know from a device standpoint, on the spray characteristics, and we also look to understand how the patient utilizes the device. For example, if they don't insert the nasal spray far enough into their nasal cavity, they may not be able to achieve, deposition in the olfactory region. And so we work, to try and provide those guidelines to customers that are using our platforms. And then we start to think about, in the nose to brain space, whether or not we need to utilize a permeation enhancer to open up some of those tight junctions in the olfactory epithelium, whether or not we need to use a mucoadhesive agent to keep that drug retained in the nasal cavity, or in the, either in the trigeminal nerve or the olfactory nerve region, to be able to facilitate that transport across the epithelium and along the nerves, or along those neuronal pathways. And sometimes people also think about nanoparticulate systems for targeting the CNS through the brain. And so that all comes into play in how we, select our devices and formulations. And so we have a range of different types of device platforms at Aptar Pharma. We have multi-dose devices. So I'll start down here. Where you can deliver, 30 or 60 or more doses. And we have modified one of these multi-dose devices. And I have a slide coming up on the next part of the deck here that talks about how we've modified that device to further target the olfactory region. And this is an example of how, using our AeroNose nasal cast, how we've been able to take this standard multi-dose pump. And so the green areas are the deposition areas. So our standard pump, and how you can increase the deposition into the olfactory region in this model using our modified pump. But we also have single dose and unit dose systems used commonly in like emergency treatments like the Narcans and the epinephrines, but also, an option for delivery to the CNS. Preservative-free pump. This is the example of the pump that Suzanne Kraft is using in her studies. So this means that your formulation doesn't need a preservative. We also have nasal powder platforms. And while not relevant for today, we also have some vaccine devices. And so we have two options really where we've looked to try and optimize deposition in the olfactory region. So this is our targeted device for nose to brain delivery. We've modified the actuator to have some functionalities to help minimize any sort of variability that the patient may have. So this helps control the depth of insertion and alignment with the nasal valve, to be able to have that spray penetrate deeper into the nasal cavity towards the olfactory region. And then using our nasal cast, you can see where we have present and deposition, looking at different regions in the nasal cavity. And so if we just look at the olfactory region, our standard pump compared to our nose to brain targeted VP7 pump, and you can see here with the gray bars that we are able to increase deposition in the in vitro model by about 40%. And so we think that's going to enable delivery into the olfactory region. As you can see here, we still have drug, the gray bars, we still have drug depositing in the turbinate region, which may also facilitate uptake via the trigeminal nerve in the event that the drug is following both pathways, olfactory and turbinate transport. And this device is entering clinical trials in early 2025. And so we're excited to see how this performs in man. And then recently, Aptar has acquired a company called Sypnose. They're an Israeli company that have a unique delivery system for nasal administration to the olfactory region. They use a little pressurized force behind the spray to drive the formulation into the olfactory region. And this is an example of a proof of concept study that they had conducted using intranasal topiramate. Topiramate is typically used for seizures, but it has been shown to be effective for binge eating. So the oral formulation is sometimes prescribed off-label for binge eating disorder. But one of the challenges is that the dose that needs to be delivered orally is quite high, and then sometimes patients will, stop using the formula or using the product because, the tablets, because the dose can be associated with, neurological side effects, dizziness, or cognitive impairment. So what SIPNOS wanted to do is to look at the nasal route for administration of topiramate. So they did a proof of concept study in 12 patients where that had binge eating disorder, and they monitored the patients for 2 weeks to determine their baseline binge eating episodes. And so they were asked to record whenever they had the urge to eat or an actual binge eating moment. And the idea for 8 weeks of the study were the volunteers were to administer the topiramate as needed. So only dose themselves when they felt they were having an urge to eat or an episode. So this was really like PRN administration, not a daily administration. And in that study, the starting dose was 60 milligrams, and they had the volunteers had the options to escalate up to 180 milligrams of topiramate. But throughout the study, there were about 500 occurrences of either binge eating or the urge to eat. And about 80% of those were controlled with the 60 milligram dose of topiramate. And then you can see here in the graph, looking at the mean number of event days over the baseline 8-week period and then a follow-up period, the study indicated that they had significant decreases in the urge to eat events compared to the follow-up period, and a significant decrease in the binge events compared to baseline. So even though this was a proof of concept study, it's an exciting opportunity to see this alternative, this new device that we now own at Aptar, being used for a CNS indication. And then finally, I just wanted to conclude with 2 things that Aptar is trying to do, and that, many people come to us, customers come to us and ask us, will our drug get into the brain? And so one of the things that we're actively trying to do is look to build a physiological PBPK model for trying to quantify transport of drug into the brain. So here's a small snapshot of what some of the parameters in that model would look like. And in order to help facilitate building of that model, because we're starting with NHPs and we'll plan to scale it to humans, we're working with various universities to develop cellular models where there are growing cell lines that have like neurovascular units and other important structures related to the olfactory epithelium. And we'll look to do transport studies to try to understand the mechanism, what molecule and formulation properties are important, and then use that to help develop and then ultimately validate our nose to brain PBPK model. And when one of the ways we're going to do this is to insert the cellular monolayer into the snap well into a nasal cast. And so when you spray the nasal spray, you'll be able to capture deposition as it would occur in the nose onto the cells. and measure transport. So we're actively working to try to help improve the understanding of drug transport, and also trying to ensure that our delivery systems are targeting the right regions in the nasal cavity. So it's really, an exciting time. And that Aptar is supporting our customers with devices. We also have devices that can be used for preclinical studies, which I didn't talk about today. We have a sister company that can help with the formulation and the analytical testing. That you got a small taste of that with what Proveris offers as well. And so we think it's a really exciting time to explore both nasal sprays and nasal powders for CNS indications, and are working with scientists and universities to try and better understand how drugs are getting to the brain and supporting that. And so, really thankful for the time to present today, and happy to take any sort of questions that you guys may have.